Hello, welcome from Wagga Wagga, New South Wales, Australia. This is a presentation for the 5th International Symposium on Sigma-2 receptors, and I'd like very much to thank the organisers for inviting me. My name is Mike Cahill, and I'm going to be talking about PGRMC1 and its role, I think, in neurobiology. And I think that's a certain merit in telling this story from the beginning to its end so that it gains perspective. So I hope that you appreciate that. So essentially, this is a modification of a figure that I've previously made, updated with a bit of new information, and it shows that PGRMC1 is a very multifunctional protein. We're here now because of this sigma-2 receptor link here, and we'll see, of course, there's a lot of other biology here that's relative to sigma-2, but we'll see how we progress on that. And I would rather reinterpret that figure as this one here, because it shows PGRMC1 with a really, really fundamentally important role to human biology. And I hope that by the end of this talk that you may at least believe that that's reasonable. So, before I progress, I'd just like to issue a community invitation. I'm co-editing a special edition of the journal Cancers, and we accept any additions on PGRMC1 up until the end of August. And this edition, it's not necessarily specific to cancer biology. Anything that we think is relevant to the cell biology that could be related to cancer, we will consider that as well. So it doesn't have to be a strong cancer-based publication, for instance, something on metabolism or cell motility or anything like that would be relevant to cancer biology. Okay, so I'll start telling you how I got introduced to PGRMC1, and I co-founded in 2000 a company called Proteosis AG. There's a glossy German Ministry of Research and Education publication on the highlights of proteome research, and there am I featured on page 38 with about 20 other of Germany's at the time best proponents of the art of proteomics. And basically how we got into this was developing what we call proteotope, which is a multiplex radio imaging system. We used to label proteins, clinical direct proteins, with iodine, either 125 or 131, to have a difference in half-life of 8 days versus 60 days. And so you could take one exposure on a phosphorimager, an imaging plate, you could leave it to decay a certain time, take the second exposure, and then knowing just simply without disregarding the half-lives, we can put those two images together and solve simultaneously for every single pixel and work out what the original signals. So we can deconvolute the signals from each isotope out, and that would give us a dual colored image. And this turned out to be very sensitive and very accurate. And so we were able to get very reproducible measurements of protein abundances in the early 2000s. So we designed our experiments on for instance, the fluorescent dye nucleic acid arrays that were around on the time. So we had 3 samples labeled with 125, 3 with 131, and we inverse label them and generate basically 6 different measurements for each sample, and that gave us statistically robust data most of the time. So it's not a bad system. Here we've showed that we can use it for laser capture microdissected cells from breast cancer. And in this is another study now where we pointed it towards estrogen receptor positive versus negative tumors. And basically we've got 54 centimeters isoelectric focusing here that was also part of proteotope. There was much larger isoelectric focusing than was commercially available. But anyway, we identified these 3 spots of PGRMC1. So here we can see the statistics. Estrogen receptor positive is blue and negative is orange. So you can see we got some keratins and things like this. And we knew from the messenger RNA analyses that luminal breast cancer cells would express these. And so that's kind of what we're getting. The estrogen receptor positives are luminal cells, and the basal-like cells would give us these things, vimentin, fibrin, etc. So we were getting basal-like cells in the estrogen receptor negative. So that told us that what we were measuring was actually accurate. And we also got some increased serum proteins in the estrogen receptor negatives. And this is a reflection of the increased edema of the infusion of serum into the tissue. And that's something that messenger RNA arrays didn't pick up. So although there's, what is it, 35 proteins in this list, but still it told us that we're getting accurate biology. And among these 35 proteins were 3 different spots of PGRMC1. And we can look at here, we've got sort of, if we go in and say this protein here, this is actually a spot of albumin, and it's what's this one? Let's go for a good one. This one here is about 2 to 1, the ratio. So the abundance ratio is about 2 to 1, and we're getting a differential, a significance there of 0.0006. So you can see there's very small amounts of protein difference could be detected very highly significantly. So at the time, I think a lot of people were publishing lists of proteins, but they didn't have this this absolute high sensitivity and high precision way of measuring. And so we could afford to hang a lot more onto these differential results. So we chose to pursue PGRMC1. It seemed interesting. And we did huge bioinformatics assays of all of these different proteins here. And it turns out that PGRMC1 is a cytochrome B5 domain. It had a few reported phosphorylation positions. One was at serine 57, and that was here near the N terminus of the cytochrome B5 domain. One was at tyrosine 131, which was 139, which was kind of in here. And another one was at tyrosine 181, which is just a little bit past of the C terminus of the cytochrome B5 fold. Now, this is modeled on an Arabidopsis protein. So although the Arabidopsis protein doesn't actually have this region of the protein, we nevertheless can say that in PGRMC1, it must be around here. So this immediately suggested that we had 3 phosphorylatable motifs that were all on the one surface of a protein. And so if you had regulated interactions with any of these 3 motifs, they would be able to cross-interact with other proteins there. And of course, if you concentrate things like this, that's increasing enzymatic activity. And so this is the way, for instance, that tyrosine kinase receptors activate their signaling. So that was a very interesting prediction just from that. We then set about to mutate some of those sites, and we thought this cysteine was interesting, for instance. And you can see that I've got set up a couple of double mutants down here. The tyrosine and the serine 180, the serine, both serines together, and the I guess that's enough. And we then put those into cells and assayed them. So there's a stably transfected HA detected variant. And essentially, the one that gave a result was the double serine mutation. So it's knocked out this serine and this serine, thereby exposing the tyrosine 179. So in this one, the cells basically became indestructible. You can't kill them with 50 micromolar peroxide, and with peroxide treatment they failed to activate ACT kinase. So there's something going on here. Okay, we also found in that same study that PGRMC1, just doing kind of the background checking, that PGRMC1 in an estrogen receptor positive tumor, the estrogen receptor was expressed on the outside of a tumor, and near the necrotic core where it gets hypoxic, PGRMC1 was induced in a ring around here. So as and it turned out that the estrogen receptor is turned off in that ring. So there's an induction process that goes on. And in the adjacent slide, we could show that this fluorescence in here is actually autofluorescence. But we have in an estrogen receptor positive tumor that estrogen receptor is downregulated and PGRMC1 is upregulated in the hypoxic zone surrounding the necrotic core. So that's the kind of situation we've got there. And this is interesting, of course, because it's this hypoxic cells that often give rise to metastases and etc. So we then looked at the induction of GLUT1, glucose transporter, which is necessary, of course, for hypoxic biology. And very interestingly, PGRMC1 was induced in some cells prior to GLUT1. So GLUT1 is induced as part of the classic HIF1 response, where HIF1 is destroyed by oxygen and stabilized in hypoxia, and then turns on its genes, including this GLUT1, which then pumps glucose into the cells so they can increase their glycolysis. So this suggested to us then that PGRMC1 could have a role in Warburg metabolism of cancer cells. We also published this model where PGRMC1 is kind of part of a central nexus signaling system where it controls cholesterol, mevalonate pathway and cholesterol signaling, leading to increased things that cells do when they've got cholesterol, as well as membrane trafficking functions, which could control the cell surface expression of various important proteins, including the GLUT family. And Rolf Craven has since actually confirmed that this is true. PGRMC1 is responsible for the translocation of the insulin receptor and glucose transporters to the cell surface. And then we proposed anti-apoptosis, endothelial mesodermal transition, cell motility, contact dependence, vascularization, and hypoxic survival. And actually every single one of those question marks has since been confirmed. So I relocated with this information to Australia in 2008, thinking that I was going to further this fantastic research proposal. I came to a regional university, and apparently the Australians don't believe that good science can happen at regional universities, because I've had 30 unsuccessful grant applications, and I'm actually, this is, as it turns out, this is my final day working in academia. I've accepted a voluntary redundancy, and I'm going to retire unless somebody offers me another job. So nevertheless, it's now demonstrable that my initial applications would have been spectacularly successful in Australia, and I hope that I'm actually going to be able to publish these for the record and show what a dismal job the Australian grant system did of supporting research that could have been really important. And I think that it's just a case of regional discrimination, but that's for another forum. So this is a figure from one of the applications that I sent actually to the Komen breast cancer program in the States. And my point was to show that I was somebody in the field. And we can see if you do PubMed and cancer, in 2003, there was one publication. In 2005, there was another couple here. And by the time the first 6 publications were out, Proteosis already had 3 patent applications related to PGRMC1 and phosphorylation. So the point here is that I think at Proteosis, we were really way ahead of the ball game. And if my grant applications here had been funded, then there would be a trajectory way up in the stars somewhere for PGRMC1 research now. Anyway, after no grant funding in Australia, 12 years later, we finally got to publish some of the results. So, I mean, my initial grant application proposed 15 mutants, and there are 3 in this publication. But nevertheless, two in this publication. Nevertheless, we see that we've got serine 57 and 181, the double mutant, and I failed in the original Neubauer to combine the double mutant with the tyrosine 180 to show that the effects were due to tyrosine 180. So this time we've gone into Mia-Packer pancreatic cancer cells. We can see that all of these proteins, it's got a HA, so it runs a bit higher. This is the endogenous protein. So we're expressing about equimolar with endogenous protein. And the first thing you notice is that we have a transformation of cell phenotype. So we've got this fibroblast looking cell type for the Mia-Packer parental cell line. The wild type expressing cells also have this elongated, predominantly elongated, I should say. There's still a couple of round ones floating around there. Whereas the double mutant and the triple mutant cells adopt this rounded phenotype. So Partho, the PhD student at the time, he assumed that this would be related to the actin cytoskeleton. And so he has used a Rho kinase inhibitor. And by doing that, he's shown that the shape change is dependent upon Rho kinase in double mutant and in triple mutant. It's a little bit toxic in triple mutant, but nevertheless you can see that, and the control cells had no change in shape. And so we have an actin-dependent process. PGRMC1 is affecting the actin cytoskeleton. We did a scratch assay and we found that the double mutant cells migrated much faster into the center of this, this scratched void zone than any of the other cells. You can see n equals 12 there, and here's the double mutant is much highly significantly different to all of the other cells. Eva Goldis, bless her heart, she funded a proteomic study, which the grant agencies wouldn't do it, but Eva found some money somewhere. And we've got this, our cell lines here. Now, what I haven't said is that each of these double mutant 1, 2, and 3, that each one of those is a separate stable transfection. So we have no clonal type artifacts. If we see an effect coming through this design, it's due to the construct and not to a cell type clonal artifact. So here we can see that the Mia packers and the double mutants have all these blue proteins here. The triple mutants and the wild types have all these red ones here. And so just alone, these mutations are clustering together without us doing anything. The proteomics clustering software is recognizing that a triple mutant is a triple mutant, a wild type is a wild type, a double mutant is a double mutant, etc. So then we, and this is 243 highly significantly different proteins. We then did a pathways enrichment analysis on these, and you can see we've got glycolysis over here and amino acid, citric acid cycle, and over here we get the things like smooth muscle contraction and mitochondria and chaperones and all sorts of things, ubiquitination over here. So I'll just look at a couple of these. One is glycolysis. So that's, of course, was very interesting to us because we predicted that PGRMC1 would affect glycolysis. And actually, the response here with fatty acid, the synthesis and catabolism, it looks a lot like an insulin glucagon response. But that's an interesting feature. We have a little pattern here that says actin cytoskeleton. We were interested in that because of our altered motility and our altered cell shape. So that seemed to be kind of on the money. And then we had all of these blue ones here. These are predicted to be PI3 kinase signaling, so PI3 kinase ACT pathway. And another prominent feature was that all these black ones here are mitochondria. And you can see that most of the mitochondria are in this red region here, which is on in triple mutant wild type and off in double mutant and the parental cells. So let's just look at the valification of those. So the ACT pathway, this is then reverse phase protein arrays, and bad serine 136 is an ACT substrate where we mutate tyrosine 180. Its level of phosphorylation goes down, whereas the bad serine 112 is not an ACT substrate and it's unaffected by the mutation series. Similarly, GSK3, glycogen synthase kinase 3, is one of the famous ACT substrates, and we can see that in the double mutant, its phosphorylation level goes way up, and it's then abrogated again by the mutating the tyrosine. So the difference, once again, between the double mutant and the triple mutant is only the oxygen phosphate acceptor on tyrosine 180. So that's what happens when we go from double mutant to triple mutant. We've taken away the oxygen from tyrosine 180. We looked at glycolysis, and we can see here's the Mia packer parental cell line. These actually haven't been stably selected. This is just the parental cell line. So the actual comparison is down here, where we've got the wild type, the double mutant, or the triple mutant. So the wild type, the more glucose is taken up, the more Warburg effect is going on. Okay, so this is glycolysis in the presence of oxygen, and in our system, the wild type gives this basal level. When we mutate those two serines and free up the tyrosine 180, then we induce a more mitochondrial active phenotype. And when we mutate tyrosine 180, then we increase Warburg metabolism. So not to the same level as the parental cell, but that hasn't undergone hygromycin selection. And so the actual control structure is in these three here. So this is glucose uptake. And when we look at lactate secretion, we get effectively the same result. We looked at mitochondrial function with a seahorse assay, and we get specific mutant type effects on mitochondrial function, but it's not as simple as saying that turn glucose off and the mitochondrial oxidative phosphorylation goes up. So there are other effects. There's a polyfunctional effect going on with the mitochondria. Nevertheless, things happened. So Sabir has just shown that PGRMC1 induces Warburg metabolism. He actually used my my published hypotheses. I actually thought that I was going to have this stuff published when I published that, but all glory to him because he got it out first. And he's shown that PGRMC1 is responsible for progesterone-induced aerobic glycolysis and that it associates with hexokinases. So this is very strongly confirming the idea that PGRMC1 is involved in Warburg metabolism that we predicted in 2018 and 2016. So we then tested mitochondria again, and here we're looking at MitoTracker, which actually binds with higher affinity to mitochondria with high membrane potential, so the proton gradient is strong, and basically without explaining the whole cell, you can see that there are very strongly significant differences, and so we have effects going on with mitochondria. So our mutations are strongly affecting mitochondrial function. We then looked at mitochondrial form. This is an image. The red is phalloidin, so it's staining actin filaments, and the green is mitochondria. It's a mitochondrial marker, and we see the parental cells. They have these long spaghetti-like mitochondria. The wild type cells also have spaghetti-like mitochondria, but they have a few of these shorter ones as well, whereas the double mutant and triple mutant are predominantly these kidney bean or green pea shaped mitochondria. So that's a dramatic effect on mitochondrial shape, which could also have to do with why the seahorse analysis was a bit more complicated. So this is what an actual analysis looks like. Carlos Casano, he went through all of these numbers here, 86, 16, 79, and annotated every single mitochondria that was detected and measured it in terms of periphery, length, and area. And he summed these for, it's a really huge data set. And I'll show you the result the next time, but I'll just show you that the populations exist between elongated and round, and they were scored blind. So somebody had to say that's elongated or round, and then they would score the mitochondria separately. And I'll just point out that the triple mutants, out of 91 cells, one of them was an elongated cell, and 90 were scored as round. So the results look like this. Basically, we get elongated and round. Mia-Packer wild-type double mutant, triple mutant. If we look at average perimeter, that's how far around the mitochondria, and then area, and then we can use these together to calculate a factor called form factor, which basically is a measure of elongation. And the longer the form factor, the more spaghetti-like the mitochondria are. And we can see that for the Mia-Packer cells, most of them have elongated shape and elongated cells and filamentous mitochondria. And this is very interesting for all the cells between 2.6 and 2.2 form factor. There's like a switch. You go from being an elongated cell with long mitochondria to a round cell with short mitochondria. And just notice there that one elongated cell that we saw in triple mutant is the one that has a form factor above 2.6. Anyway, we seem to have some sort of a relationship between cell shape and mitochondrial form, and this is related somehow to mitochondrial metabolism. So this looks like a fairly fundamental eukaryotic property being altered here. Not really relevant here, but well, it is to cancer researchers. But if we took out the tyrosine 180 from PGRMC1 in our mutants, then it totally abrogated their ability to efficiently grow tumors in mice. And should remember that the endogenous protein is in there as well. So it's a wild type protein, but this protein here just has tyrosine 180 mutated to the phenylalanine, and it's unable to grow efficient tumors. So that's fairly striking. We've done nothing else to the cell except put in tyrosine 180. So it's interfering with tumorigenic mechanisms fairly strongly. So all of these effects on mitochondria made me wonder whether PGRMC1 may have been involved in mitochondrial origins, that is, in the origin of eukaryotes. And there's a hypothesis here. I spoke about this last year. You can look it up in the previous archive talks if you want, but that the mitochondrial regulating properties of PGRMC1 may have come from eukaryogenesis, and it's involved in detoxifying newly emergent molecular oxygen for an Asgard Archean proto-eukaryotic host cell. So the first thing to do is to look where might PGRMC1 have come from. So we've got lots of scanning prokaryotic sequences, and it turned out that there are two types of cytochrome B5 domain we found. One here with lots of bacterial proteins in there, and the eukaryotic cytochrome, so the cytoplasmic and mitochondrial cytochrome B5s, they're in this thing called clade1 here, which is separated in this analysis with 94% bootstrap strength from this clade2, which has also lots of bacterial. And in this particular analysis, there's no archaeal sequences, but they also have clade1 and clade2. But I'm showing the bacterial one because there's another separation here. And then we get this group, which includes MAPR proteins and some bacterial proteins. And these bacterial proteins don't have a me here, but very interestingly, they have the tyrosinate heme chelating residues of PGRMC1, of MAPR proteins. So we've got heme is chelated by histidine over these ones here, including some of clade2, so that would be the ancestral position. Then in this subclade here, we have tyrosinate heme chelation. And then in this clade here, we have the MAPR, the eukaryotic MAPR proteins. So I've called those cytochrome B5, the cytochrome B5M or MAPR-like. And a subset of those is the cytochrome B5MY, which is the MAPR-like with tyrosines. And these are the most closely related prokaryotic proteins to MAPR proteins. So it could be that these guys, the MYs, came from horizontal gene transfer from eukaryotes to prokaryotes, or, which I like more, and that's what I'm going to talk about, but I formally say it's not clear, that maybe that one of these cytochrome B5MY proteins gave rise to the MAPR proteins. So we can look at the separation, the sort of sequence, logo sequence analysis of this. The MAPR proteins, you can see there are our two tyrosines. There's our me here, which is this mitochondrial MAPR interhelical insert region. It's inserted between two helices here. And this is eukaryotic specific. The cytochrome B5MYs, you can see they've got those tyrosines there that are involved in heme chelation. The cytochrome B5Ms, they don't have those. They have histidine here to chelate the heme. And then if we look at the clade1 consensus, we've got kind of, well, that doesn't really matter. What's important is to go down to the prokaryotic cytochrome B5s. And we've got this thing here, this HP motif. And this H is really important because it's involved in heme chelation in cytochrome B5s, and it's present in prokaryotic and eukaryotic cytochrome B5s. Okay, so it's part of what I'm going to call loop 2, and I haven't talked about loop 1, which is present in the clade1 proteins, and it's followed by a glycine. So there's a sharp kink in the peptide backbone, and then there's a loop, and we can find that on PGRMC1. So we know what it looks like. It's a surface loop. So these are discriminating features between these different groups. So you can see just basically here and here that they're really different proteins. Okay, so essentially we've discovered a new class of prokaryotic cytochrome B5 domain proteins. And we can conclude from this that in PGRMC1, the tyrosinate heme binding is probably of bacterial origin, although that's not clear. We see here the H bonds with the propionate groups, the tyrosinate heme chelation, and hydrophobic heme interactions here. And what's important is that in the eukaryotic system, that the me here has been inserted between residues of the heme binding domain, so that one helix goes up and another comes down, and at the junction of those two helices, this eukaryotic specific thing here has been inserted. So it's highly conceivable that what happens at the me here is going to influence heme binding, and heme binding is going to influence what happens at the me here. So there should be some sort of interplay between these functions. This is, we went through and looked at the different conservations, and it turns out that to build a cytochrome B5 domain, the most conserved proteins are the ones that pack the inside of the protein. But interestingly, the proteins that are conserved in clade 1, that is the MAPR-like proteins, as opposed to clade 2, which is the conventional cytochrome B5s, form a continuous region on the surface of the protein. This is a conserved region on the surface of the protein, and so this conservation of surface would imply that there's some sort of interacting protein that must be, there's a protein interaction that must be conserved. And nothing is known about that whatsoever, and that would be a good place to look for things interesting. So we've also crystallized a cytochrome B5M protein. And we confirm here that it's got this loop 1 on the surface, so it's like a MAPR protein there. And here we can see the loop 1 over here. This is clade 2 proteins with a little tiny loop, and we've got this great big loop up here, the loop 1 here. But what's interesting over here is this is clade 1 and this is clade 2 proteins, and we can see here in the clade 2 that the propionates of the heme are kind of sticking out of the screen. Whereas in the clade 1 protein, the MAPR and this prokaryotic protein, the propionates are swung about 90 degrees around. So the mode of heme binding is different through this tyrosinate heme chelation, or through the cytochrome B5M heme chelation. And importantly, this loop 2 here, that histidine that I showed you is just about here, and it interacts directly with the ion of the heme group in here, and it's just absent here. So the heme binding motif must be different. So we've really identified a new class of cytochrome B5 proteins, and MAPR proteins arose from this clade 1 type. Whether it's from the B5MYs or from B5Ms it remains an open question, but it definitely came from there. So if it came from B5MYs, then many of these proteins are present in an operon that looks like this. So this is kind of interesting, that it has a 2-component signal transduction, so it's somehow inducible. It has 3 cytochrome B5 proteins, so heme binding, redox reaction, and it has a ferric reductase-like domain. We don't know if it's actually ferric reductase, it's just sequence homology at the moment, but this operon screams out at you, it's doing something redox related, doesn't it? And this one over here, we don't know what it does. It's not similar to anything else. So it's possible that our cytochrome B5MY proteins originated in an operon like this. And you can imagine maybe that electrons get transferred, and they're all transmembrane proteins. So we have a membrane complex, and electrons get transferred to the cytochrome B5MY. And maybe then the heme is dissociated. So the heme carries the electron somewhere else. And why do I think that? Well, it's a feature of this tyrosinate heme chelation that when we accept an electron, the heme affinity for the protein is lost. And this is the case for yeast proteins, PGRMC1 and PGRMC2, as you can read in those references there. And so this is a feature of PGRMC1 that probably maybe not properly appreciated, that we have heme bound and non-heme bound, and the non-heme bound may be available to do something else. For instance, maybe the me here, whatever it does, is available to do something in the heme or non-heme bound form. And so by dissociating heme, then we can change the functional attributes of the protein and turn something on. what else here? I had something else I wanted to say. Oh, this is a type of one-click chemistry. If this were PGRMC1 and it accepts an electron during a cytochrome P450 reaction, then and the heme dissociates. There is still an affinity, so it doesn't automatically have to dissociate, but it could. And if it did so, then that would mean this kind of a one-click chemistry. When we activate sterile synthesis, for instance, maybe that also sends PGRMC1 off to do something else after it's performed that reaction. So what's this got to do with eukaryogenesis? Well, eukaryogenesis occurred around 1.85 giga years ago, or billion years ago, or after that, so not before that. And this is after what's called the Great Oxidation Event, which was the evolution of photosystem II by bacteria and the accumulation of oxygen in the atmosphere. Now, this was a catastrophe for our forebears, our archaebacterial forebears, because they were anaerobic and they couldn't survive. So many people think that the advantage of mitochondria is because they can make 32 ATP instead of 2 ATP. But that's actually not the case, because reconstructions of closely related archaebacteria, particularly by Ts. etama, have shown that the archaebacteria would have had its own electron transport chain, was perfectly capable of producing ATP. So the advantage of the endosymbiotic event, the capture of the mitochondria, was because the mitochondrial electron chain used oxygen as the electron acceptor. So in other words, if we feed the mitochondria, we can neutralize oxygen and survive. And this turns out to be a key feature of eukaryotic biology. So PGRMC1's role in this, very interesting. In lots of bacteria, you have this thing called a hopanoid synthesis pathway, where the mevalonate pathway produces the isoprenoid squalene, and an oxidosqualene cyclase then cyclizes that into a 5-ring structure. This is not shown here, but it's called a hopanoid, a 5-ring structure, which serves analogous functions in bacterial membranes as cholesterol does in ours. So what eukaryotes do uniquely, and all of them, is when they make steroids, there's a new enzyme comes in, this squalene epoxidase, which makes this epoxy squalene derivative here in an oxygen-dependent manner. So if you want to be doing something to protect yourself from oxygen, you need an oxygen-dependent reaction to run that. And so that's what's going on here. And this then is the substrate for the eukaryotic oxidosqualene cyclase, which produces lanasterol in yeast and humans, the first sterol that's produced. The very next reaction involves cytochrome P51, the most highly conserved eukaryotic cytochrome P50, and PGRMC1. So it is the 14-demethylation of lanasterol to make this molecule here, which is also a meiosis activating sterol in mammals. And that's very interesting because meiosis was a eukaryotic invention. Anyway, PGRMC1 is right here at the root stem. And of course, the reaction involves oxygen. And so we have 2 oxygen-sensitive reactions. And this is just the type of system we would want if we wanted to regulate the mitochondrial activity in a endosymbiotic organism. So this is then a hypothetical model of what might happen in eukaryogenesis. We've got our cytochrome B5MY, which accepts an electron while lanasterol is demethylated. The heme dissociates. We have the lanasterol, which now could potentially bind to the empty pocket, and that may then be related to PGRMC1 sterile sensing. Alternatively, this might go and do something else, and that would then presumably activate or deactivate, whichever the case may be, the mitochondrial metabolism. Presumably would upregulate it. I'm not thinking straight there. Okay, so that's a model for how PGRMC1 is involved in steroidogenesis, in eukaryogenesis. So interestingly, we've got here the mevalonate pathway, squalene, lanasterol to FF-MAS. This is the cytochrome P51 reaction. And just as a sort of interesting thing, that all of these subsequent steps down to the synthesis of cholesterol must have evolved after this reaction here. So this is really the first reaction, okay, the first steroid modifying reaction in eukaryotes. So that places it right down there near the base of the eukaryotic evolutionary tree. Very interesting for me that Sabir has also shown in one of those papers that I talked about before that the contacts of mitochondria with the endoplasmic reticulum at the mitochondria endoplasmic reticulum associated membranes, the MAMs, this can be regulated, not in all cell types, but in some cell types by PGRMC1. So this would be a great, if we think about the eukaryotic situation where genes for lipid synthesis have been lost from the symbiont and they're now present in the nucleus, so that the lipids are now made in the endoplasmic reticulum, but they have to get back into the endosymbiont. That has to be regulated. And of course, PGRMC1 is just with this membrane trafficking, sterile synthesis. It's just the right type of protein. It smells right to have been doing that. And this is only a preprint at the moment. Let's hope that it stands peer review. But for me, this is a very exciting finding that PGRMC1 can regulate this MAM interaction. Okay, so a hypothetical summary so far. And why is it hypothetical? Because I haven't had a grant in 12 years, and a great way, Susan in Catalano introduced me at one of these talks as a great hypothesis maker in the field. How do you take a great research scientist and turn them into a hypothesis maker? Don't give them any money. Anyway, so proposed ancient PGRMC1 functions include steroidogenesis, tyrosine heme chelation, this one-hit chemistry, and sensing of something redox, regulating mitochondrial oxygen consumption, which is related to yeast sterile synthesis and Warburg metabolism, hypothetically, and the membrane trafficking may be related to the eukaryotic acquisition of the media, and why I'll show you. I'll show you later. So then we go to eumetazoan evolution, and PGRMC1 phosphotyrosines coincide with the appearance of the gastrulation organizer. This is, for my money, the hottest thing in PGRMC1 biology at the moment. So we're now jumping forward from eukaryogenesis up to the origin of animals, which is about 1 billion years. And during this time, this group, the epistheconts, including yeast and humans, they evolved, and a certain group of those called the holozoans, they started developing things like sarcomology 2 domains, tyrosine kinases, and lots of other animal-specific or animal-required proteins, brachyury signaling, brachyury actin cytoskeleton induction. So I was interested in these because I wanted to see when these PGRMC1 tyrosines came around, and I was kind of thinking that probably some stage early in the protist, the single-celled evolution, but it turned out that's not the case. It turns out that we've got this basically the PGRMC1 cytochrome B5 domain was short, like a bacterial one at the end, the C terminus, up until we've got this tenophorin, but especially the placozoan. It's a form of animal between sponges and higher animals, if you like. And then we've got the cnidarians, which are things like sea anemones and jellyfish and hydras and things like that. And then we've got the bilaterian animals. Now, bilaterians, just so you understand what this word means, things like mollusks and insects and nematodes and annelids and chordates, all of those things that are bilaterally symmetrical. So they all have this motophore. So PGRMC1 at this stage has gained a C-terminal extension, and in that C-terminal extension, at the stage of the cnidarian ancestor, it's the ancestor that is common to bilaterians and cnidarians, and these are called eumetazoans. So these have tyrosine 139 and tyrosine 180. The cnidarians also have a stretch of negative amino acids with a phosphorylatable residue in there. When we get to bilaterians, we have cemented in a phosphorylatable residue, a gap, the tyrosine, an adjacent phosphorylatable residue, and then some negative amino acids. So this is conserved among all those animals I just mentioned. Some of them have actually lost it, but it's strongly conserved, so it must have had something to do there. So this actually also corresponds with the gastrulation organizer. So this Loimka, the last eumetazoan common ancestor, was the first animal that actually had gastrulation organizer. And the importance of that is the gastrulation organizer leads to the subsequent differentiation of different cell types. So before that, you've basically got some epithelial type cells and sponge and placozoans, and not too many different cell types going on. But once we have a gastrulation, then we start to see a proliferation of different types of cells and more specialized animals. And this placozoan, there are some people who actually think that it may have actually once had an organizer and lost it secondarily. And that's interesting in terms of it's got this extension here, but it's lost the tyrosines. And if that's so, then it would implicate these tyrosines as being important in organizer function. Not to worry too much about that. So the timeline of this, we've got here hundreds of millions of years, so it's essentially a zoom in on what I showed you before. And we've got the branches of the sponges, the placozoans, the cnidarians. So this cnidarian here, this is where the loimca came, and the last eumetazoan common ancestor. And it's basically corresponding to what's called the Sturtian glaciation. This is the largest snowball ice Earth event that was ever happened. So there have been a couple of other smaller ones, but this essentially, the Earth's oceans are thought to have frozen over, and that would have radically changed living conditions. So in response to this, animals developed gastrulations. Very interesting thing. And then the bilaterians appear by this time here, still in the Cryogenian, where these iceball Earth events happen. And by the time the first animal fossils appear in the Ediacaran fauna, you can see we already have bilaterally symmetrical animals and cnidarians already present. So this organizer, also the Netron DCC system event appeared in this loimca, as did the concept of sleep. So they're also the first organisms to have neurons, as we'll see. And Netron DCC is important because PGRMC1 interacts with DCC and its homologs across different phylogenetic groups. So the Sturtian glaciation, we can see that although the oxygen levels are generally going up, the Sturtian glaciation led to a crash in oxygen levels. So this was a real, if you like, a redox challenge to organisms here. It's actually named after the same Charles Sturt that my university is named after. So this is the Ediacaran fauna here. It's a reconstruction, of course. We can see there are jellyfish, there's a cnidarian, so we can see these little bilaterally symmetrical things swimming around. So all of those animals reconstructed from fossils would have had PGRMC tyrosine 139 and 180, and presumably they're important in generating those body forms. So what's going on here? Well, if we look at schematic development, the closest single-celled organisms to us, the choanoflagellates, they can form, some of them can form sheet-like coalescences, which are essentially like an epithelial cell layer. And then through the action of adherence junctions, they can form a sphere. So choanoflagellates can form spheres, which kind of look like the blastocele. What's characteristic of animals is that one cell divides to form this blastocele, a sphere, a hollow sphere full of, well, a hollow sphere of cells. And then we get gastrulation. And in gastrulation, once again, it's the adherence junctions which mediate via the actin cytoskeleton. They mediate the invagination of this feature here, which in cnidarians forms this gut cavity here, and that matures into what is essentially, these are just two epithelial layers, one on the outside, one on the inside, with this sort of extracellular matrix stuff called mesoglia in the middle of them, which can be thick or thin depending on which organism you look at. So it turns out, it's recently emerged that the cnidarian endoderm is actually homologous with our mesoderm, and the pharyngeal ectoderm is homologous with our gut. So it has things like insulogenic secreting cells, similar to the pancreas, for instance. So during what happened with the bilaterian evolution is that these adherence junctions in what's called the epithelial mesenchymal transition, the adherence junctions loosen and the cells adopt a migratory phenotype, and they can then migrate out of the epithelial layer and into the interior. And this provided the building blocks for cells to then be able to, for animals then to be able to produce internal organs and vascularization systems and et cetera, and become us. So this is a really important step, and it's just my depiction of what the first bilaterian animal might have looked like. It's thought that it might have had a slit-like gut with two openings, and in insects, for instance, this one became the mouth, and in us, this one became the mouth. But anyway, it's not so important here. The actin cytoskeleton. So the invagination of gastrulation is driven by epithelial adherence junctions. The epithelial mesenchymal transition associated with brachyuria-induced changes to the actin cytoskeleton. And we saw previously that PGRMC1 alters actin cytoskeleton and cell mobility. So the adherence junctions, they kind of link along here, and by coordinated contraction between various cells, you can change the shape of an epithelial. So for instance, if you think of a jellyfish, it's really just an internal and an external layer, and it's been contorted into that jellyfish-like shape by the action of adherence junctions during its embryology. So adherence junctions, we've got two cells here, and we can see there are vinculins and actin fibers cross-linked here, and actin filaments. So vinculins, vinculin was upregulated by the PGRMC1 double mutant, and its knockdown, knockdown destroyed the ability of those cells to migrate. So that's in Thajir et al. There. And the alpha-actinin was the most significantly differential PGRMC1 associated protein that was differential with AG205, the presumed PGRMC1 inhibitor, in a recent publication that we made. So this is right in terms of PGRMC1 biology. This is just from this Cahill et al. We can see that PGRMC1, this is plus or minus AG205, and we can see that wherever it says CH, that is a calponin homology domain, which is an actin binding domain. And this little blue thing here is it means that it's involved in the actin cytoskeleton. So of the top most significant differential proteins that we found, most of them were associated with the actin skeleton. Okay, one of them is a myosin there, for instance. Okay, and we validated two of them in that particular publication because we didn't have any resources to do any more. So interestingly, in that context, there's a motif in the me here that's predicted to form a coiled coil. And this motif is similar to motifs found in many myosins from different organisms. So there's the line up there. And this is actually tyrosine 139, which is one of the tyrosines that we gained at this last eumetazoan common ancestor. So coiled coils, in case you don't know, they required a heptad repeat of hydrophobic residues to align the two coils next to each other. And one of these is tyrosine 139. So obviously, if tyrosine 139 would be phosphorylated, then you're going to break the ability to form a coiled coil there. And you're also going to gain the ability to recruit a sarcomology 2 interaction. So this is a real fundamental acquisition of function by PGRMC1 at this stage, and it correlates with neurogenesis and the gastrulation organizer and all of those things. So absolutely, I'm tipping that this is involved in that. There we can see the motifs in PGRMC1 and 2. Interestingly, we've got coiled, strong, and weak. So this part here would be predicted to coil, and this part here would be sort of flapping in the wind a little bit. And we can see here, for instance, in myosin 10, here's the motif, and we also have strong coiled tendency here, and this part here would be flapping in the wind. And these parts that flap in the wind, they're thought to be available to make protein interactions with other proteins in coiled coils. So the thought is that maybe PGRMC1 is interacting with some of the same things that all of these myosins do. In other words, it's interacting somehow with components of the actin cytoskeleton. And I showed you that in our pull down assays, we identified actin cytoskeleton components as the most highly significantly affected proteins. So what's going on then? We can see that we've got this me here, which was acquired by eukaryotes, but now at the last eumetazoan common ancestors acquired this tyrosine, which enables its phosphorylation to recruit SH2 domains, and that would abrogate myosin coil-like interactions with presumably elements of the cytoskeleton. We also have a tyrosine acquired on this new C terminus, which didn't exist before, and we've got this little TVYS with a couple of negative charges up here, this motif here, which seems to be important, and we would predict that this regulates PI3 kinase, etc. So the Loimka had these two tyrosines, and the bilaterians have very firmly cemented in this motif T178, Y180 and S181. That should be 181, sorry, S181. Okay, if we think about all that, well, what is the actual complexity of the Loimka, the last eumetazoan common ancestor? It wasn't actually too bad. So we've got this process here, and we developed this thing. It's essentially two myoepithelial layers with some secretory cell types, photoreceptors, and neurons. So if we wanted to work out what was going on, it's probably not so difficult. And here we can kind of see what's the structure. We've got two myoepithelial layers, a bit of mesoglia in between, and that's on the next slide here. We can see this would be then the gut in here. This is the ectodermis. Most striking thing for me is the appearance of these neurons. This is a new cell type that animals didn't have before. Okay, so they've got axons, they've got synapses, and they've even making axons between the two cell layers so the organism can interact. This muscle is important here. It's basically using the adherence junction, but now we have a ring muscle and a longitudinal muscle. So that means we can get a peristaltic motion type going on. So this is real muscular contraction now. So whereas we think of an organism that might be able to bend its shape with adherence junctions, that's totally different than the swimming motion, for instance, of a jellyfish with that bell shape. Bloop, bloop. That is peristaltic motion made by muscle contraction of these muscle fibers here. So this is a fundamentally new invention. The architecture is different than our muscle, which is of course mesodermally derived and doesn't exist in this body grade yet. But nevertheless, the gene repertoire is there, present in the common ancestor of cnidarians and us. And the other thing is that to notice is that all of these cells are in contact with each other. So whatever PGRMC1 is doing here, it's able to mediate cell-cell contact type stuff. We don't need any long-range communication other than via these neurons. So if we want to take an organism like that and make an organism like us out of that, how would we go about doing that? How would we turn a Loimka nerve net into a human central nervous system? So one thing that this is also a slide I used last year, but I'll just give it again to remind you that during neural differentiation of the mouse, we have a population of neural progenitors, radial glia cells here, and they're exhibiting glycolytic biology. It's driven by HIF1. And at this stage, HIF1 is active because there's no oxygen around. As the vascular system, the vasculature develops, it brings in oxygen, which leads to the degradation of HIF1. And under this environment, we get neural differentiation. So neural differentiation is dependent upon oxygen. And I went to great pains to show you that the oxygen tension changed very much at the same time as animals evolved, and that probably we're still kind of looking at the development of these in our embryology, a mirroring of that process in our embryology. So we have this HIF1, which found in placozoans, and PGRMC1 must be involved in here somewhere. How is not clear. So if we then jump forward and we have a complex human brain here. The oldest nerve in this is the vagus nerve, which goes to the tongue, larynx, and the parasympathetic innervation of the viscera, etc. So the vagus nerve. This shows us what it innervates. And if you like, the vagus nerve must be an evolutionary remnant of those initial contacts between in the loimca between neurons and muscle cells and whatever else is being coordinated there. And what's happened is the nervous system has expanded and become centralized, but it's built on the same premises. And so I'm proposing that PGRMC1 will have important functions in the vagus nerve. Of course, the next thing that happened was that actually the centralized nervous system expanded in humans. We've got all these different organs here, and many of them have high PGRMC1 expression. So it looks like there was a toolbox that was necessary to build neurons, and PGRMC1 is part of that, and it's involved in all of these neural functions these days. So PGRMC1, as we know from cognition therapeutics work, for instance, is involved in synaptic function, and it's involved in the sigma-2 receptor complex, which, when blocked by amyloid beta oligomers, leads to Alzheimer's disease, synaptic dysfunction. So PGRMC1 seems to be at a place where it's important in synaptic function. Hormonal communication, once again, think going from that loimca where all the cells are touching and evolving this into a complex organism like us. That all happened by steps, and each one of those steps must have stretched the existing tendons a little bit further, and hormonal communication is one of those. And so we've got sort of four major axes of hormonal of hypothalamus pituitary action here. So this is the central nervous system, and then hormones. And it turns out PGRMC1 is in the pituitary, it's in the hypothalamus, the hippocampus. Down here at this end, it's involved in steroid synthesis. It's involved very strongly in reproductive. It turns out it's involved in insulin signaling. It's involved in glucocorticoid synthesis in the kidney. I don't know of reports of it being involved in a thyroid, but my guess it would be, and we could just as validly add the insulin glucagon regulation of glycolysis to this pathway as another, because it also affects the brain. Diabetes, Alzheimer's is kind of looked at as diabetes type 3, so we have a strong association here, and this is a superimposed regulation based upon blood glucose level, which never existed in our ancestor. But upon the oxygen-mediated mitochondrial regulation. So if you work your way through that logic, then all of this stuff, and PGRMC1, I haven't marked all the places where it's involved there, but lots of these cells express PGRMC1. So the hypothesis then is that PGRMC1 is pretty basic in animal biology. And the sigma-2 receptor interaction with PGRMC1 presumably evolved during animal differentiation, because it's involved particularly in the endocytosis from the vascular system, which was a bilaterian evolution. So that would be my tip, but it's actually hard because the sigma-2 receptor is hydrophobic, and so it's not very well conserved. So it's hard to find where the gene actually is. So mechanism of animal tissue differentiation, or epigenetics. Remember, I showed you that mesenchyme moving into the gastrula and forming the evolutionary raw material for evolution to build our bodies out of. Well, the mechanism that evolution adopted was through epigenetic regulation of the genome. So some genes in tissues will be on and off, and this is regulated by histone methylation, which is a very old thing from single-celled organisms. But the CPG-mediated genomic methylation, that comes with animals and is involved in the definition of bilaterian tissue differentiation. So one of the most striking things that we found, and we came by roundabout methods that I won't go into, but basically a metabolomics study led us into looking at this. And it turns out that all of those three mutants that I showed you before, each one takes the genome to a totally independent area of genomic methylation, right? So you can see that each of those 3 clones is very close to the other 3 clones, but a long way away from all of the others. This is one of the strongest and to my mind, most important biological effects I've ever seen. This PGRMC1 effects on epigenetics here, totally out of the blue, unexpected. And it turns out that it seems somehow related to the involvement of the organizer. So there's some sort of a platform here that the organizer function is built on, a subsequent tissue differentiation. And if you pull the PGRMC1 pin out of that, it seems to unravel. And this is a diagram just to show you what the strength of this is. This is the top 50 most differential CPG islands between triple mutant and double mutant. So remember, the difference between those is the oxygen on tyrosine 180. We remove the oxygen on tyrosine 180. We go from this blue one here to this red one here. So it's like night and day. So that level of genomic methylation is mediated by tyrosine 180, which was acquired at the same time as a gastrulation organizer in the loimca. Okay, so the important thing is many of these CPG islands are hypermethylated, which means that you associate that with turning the genes off, but a small number are also hypomethylated. And pathways analysis placed these with developmental pathways and cell type specific promoters. So it's almost just like the mutation of tyrosine 180 acquired with a gastrulation organizer induces a hypermethylated state resembling that of the pluripotent embryonic stem cells, because they are also hypermethylated, just like the triple mutant was. And these pluripotent stem cells represent a state prior to gastrulation. So you see what I'm saying? We remove tyrosine 180 and the cell genomic epigenetics seems to want to go back to a pre-gastrulation stage, back to a stem cell-like stage. And, you know, like, I really should be able to get a grant with this, but in Australia, it's not possible because they don't think that good science could occur in Wagga Wagga. Anyway, therefore, mutation of tyrosine 180 appears to be related to fundamental animal cell differentiation identity mechanisms. And this really needs to be examined. I can't do it if I don't get a job somewhere, but somebody really should get onto this. And the null hypothesis here that CPG epigenetics is built upon whatever PGRMC1 enabled in the loimca. And that has to do with cell differentiation processes, enabling new cell types. And the most prominent of those, to my mind, is the neuron. So PGRMC1 and Alzheimer's. So I'm just going to remind you now what I said last year, so I don't want to spend too much time on this. But Glasgow et al. Showed that the long-term potentiation requires an interaction between netrin and deleted in colorectal carcinoma, which leads to the upregulation of new components for the AMPA receptor, and then subsequent stimulation of this synapse leads to synaptic strengthening, and actin cortex gets strengthened, and all of that mechanism, and that leads to long-term potentiation by strengthening this connection. So when we have A beta oligomers binding to there, and it's shown here as binding sigma 2 receptor, but it probably binds another protein. Don't get too hung up about that. And I also don't care if PGRMC1 is post or presynaptic or inside or outside the cell. It's just a model. Okay. Anyway, that somehow the A beta changes PGRMC1. This is a hypothesis. Changes PGRMC1 function here, which inactivates the ability of DCC to interact properly. Maybe it's not cell surface localized, for instance, I don't know, which then turns off all of these things. And then the cognition therapeutic CT1812 or later abrogates this effect and leads to long-term potentiation. So that's a model that I think is quite reasonable. And maybe Warburg or pseudohypoxia could play some role in this process. So if Alzheimer's disease reflects PGRMC1 neurobiology, then we expect Alzheimer's to be associated with mitochondrial form and function, epigenetics, and glucose metabolism, and the Warburg. And basically, you can see from the literature here, cited in this article here, that that's the case. And the usual suspects, what most people spend their Alzheimer's research money on looking at tau, amyloid beta, and ApoE, or ApoE4 in particular, is a low risk Alzheimer allele. Anyway, all of these are associated with PGRMC1 sigma-2 receptor LDL receptor trafficking, and you can read that in those there. And so what I'm saying is I'm unaware of any other protein that so closely mirrors the biology, the pathology of Alzheimer's disease. So PGRMC1 to me just seems to be the metal in the blade that's causing Alzheimer's disease. Maybe I'm wrong, but it just seems really close to me. Anyway, my conclusions, which once again remain hypotheses because this work is essentially not performed, but through lack of funds. But PGRMC1 biology, which includes tyrosinate-mediated dissociable heme, steroidogenic cytochrome P450 interactions, steroid sensing, progesterone response, for instance, and membrane trafficking in actin cytoskeleton. This PGRMC1 biology is central to key eukaryotic processes involved in eukaryogenesis and the gastrulation organizer neurogenesis, ? And when we perturb this biology, then we have the ability to change processes that have been in place since those evolutionary acquisitions. In other words, we change processes that have been in place that our whole tissue identity system can be built on. And so we have the great potential to alter states of differentiation and induce disease clinical manifestations. So these processes are foundational to the origins of cell type differentiation, and in evolutionary and physiological terms, this correlates with neurogenesis, neurodevelopment, and neurobiology, which is why we should talk about it at a neuroscience society meeting. Just one caveat there, beware the buyer, that PGRMC1 knockouts are viable in fish and mice. So whatever function it exerts, it's somehow sidesteppable because you can still get mice with neurons, for instance. So that obviously deserves a closer look. And just in my own interest, before I finish, I have accepted a voluntary redundancy from my current employer, effective January the 15th, which is the day in Australia when the 14th my session will be. That's because a teaching-only role was undesirable. I'm only interested in pursuing PGRMC1 research, and apparently this is not possible in Australia. Although I'm now financially independent, I don't need to work. I'm very much available to relocate internationally and pursue PGRMC1 research if this interests you, and if so, you know what to do. Thank you very much. And I'd like to acknowledge my collaborators, especially the two PhD students who did most of the mutant work there. Bashar is just writing up his PhD now, and a special great thanks to Eva Goldis, who was able to liberate some funds to enable this work. What I've talked to you today about would not have been possible without Eva Goldis. So Eva, thank you, thank you, thank you. And thank you all for your time. I hope to be able to answer any questions you have. Okay, thanks very much. Bye.